In scientific research, cells are like tiny factories, constantly producing the building blocks of life. These microscopic entities are the foundation of all living organisms, tirelessly working to maintain the delicate balance of biological processes. Each cell is a marvel of nature, equipped with intricate machinery that allows it to perform many functions essential for survival. Scientists often need to study these factories in detail, but normal cells can be quite fickle. They can change their behavior unpredictably, making it challenging to obtain consistent and reliable results. This is where stable cell lines come in. These specially engineered cells provide a solution to the unpredictability of normal cells, offering a consistent and reliable platform for scientific investigation. By maintaining a stable genetic makeup, these cell lines ensure that experiments can be replicated with precision, leading to more accurate and meaningful findings. Imagine being able to tweak a cell's genetic blueprint, inserting a gene that makes it produce a specific protein of interest. This process allows scientists to customize cells to exhibit desired traits, enabling them to study the effects of specific genes and proteins in a controlled environment. Now, imagine that this change is permanent, passed down through generations of cells. This means that the modified cells can continue to produce the desired protein or exhibit the desired trait indefinitely, providing a reliable and long-term resource for research. The stability of these cell lines is a key factor in their utility, as it allows for sustained and consistent experimentation over time. That's the power of a stable cell line. There are two types of stable cell lines. One is achieved by eukaryotic vectors that harbor elements for episomal maintenance in the nucleus of a transfected cell. Another is achieved via direct integration of the transfected plasmid into the target cell's genome. Episomal stability is often limited, and episomal plasmid elements are often restricted to certain species. Therefore, integration into the host cell chromosome is in common use. Although integration into the host cell chromosome is a rare event, and, for most purposes, clonal events have to be isolated, the stability of the intended genetic modification usually is much higher. Major challenges for the generation of stable cell lines are low transfection efficiency and integration frequency. Stable expression can be influenced by the transfection method used. The transfection method determines the cell type for stable integration. It is known that liposome reagents can be used to transfer DNA into adherent cell lines, while viral methods or electrotransfection are used to deliver DNA into primary cells or notoriously difficult to transfect suspension cell lines. Unfortunately, viral methods suffer from several limitations, such as time-consuming production of vectors and safety concerns, while electrotransfection suffer from the low cell survival rate. This transfection process is a cornerstone of modern biological research, enabling scientists to study gene function, produce therapeutic proteins, and develop novel disease models. Transfection techniques have evolved over the years, becoming more efficient and reliable, further enhancing the utility of stable cell lines in scientific research. Stable cell lines have become indispensable tools in a vast array of scientific disciplines, revolutionizing the way we approach biological research and medical advancements. Their applications span from fundamental research to clinical applications, providing a versatile platform for a multitude of scientific inquiries. One of the most prominent uses of stable cell lines is in drug discovery, where they play a pivotal role in the development of new therapeutics. By engineering cells to express specific drug targets, scientists can screen thousands of compounds to identify potential drug candidates, streamlining the drug development process. By introducing genes into cells or disrupting existing ones, researchers can study the effects of these modifications on cellular processes, shedding light on the intricate mechanisms that govern cell behavior. This approach has been instrumental in unraveling the complexities of signaling pathways, gene regulation, and disease mechanisms. Understanding these pathways is essential for developing targeted therapies for various diseases. Stable cell lines also serve as valuable tools for producing therapeutic proteins, such as antibodies and hormones, which are critical for treating a wide range of disease. In gene therapy, for instance, researchers are exploring the use of stable cell lines to deliver therapeutic genes into patients. This innovative approach has the potential to revolutionize the treatment of genetic disorders. Stable cell lines are also being used to develop personalized medicine approaches, where treatments are tailored to an individual's genetic makeup. This personalized approach ensures that patients receive the most effective treatments based on their unique genetic profiles. In order to select stably transfected cells, a selection marker must be co-expressed with the target protein. The marker gene could be on either the same plasmid vector or a second co-transfected vector. There are a variety of systems for selecting transfected cells, including resistance to antibiotics, puromycin, neomycin, DHFR, and glutamine synthetase. After gene transfer, cells are developed in a medium containing the selective agent. Only those cells that have contained the drug-resistant gene survive. Methods to generate stable cell lines. Depending on the scope of the experiment, several options are used for the generation of a stable cell line. A mixed population of drug-resistant cells can be used directly for experimental analysis with the advantage of generating fast results, but also the disadvantage of dealing with an undefined and genetically mixed cell population. Another option is to generate a monoclonal cell line. In this method, it is necessary to dilute the resistant cells by plating in 96 well plates in such a way that culture as single and isolated cells. Subsequently, the cloning of a single cell may be repeated several times to obtain 100% clonal purity. This culture method can be used for screening experiments or conduction studies by using a homogeneous and defined cell system. Depending on the type of expression you're interested in and the construct that you are incorporating, there are many different approaches for the generation of stable cell lines. This protocol is specific for the generation of a monoclonal cell line that is resistant to antibiotics G418, neomycin. The end result that you are looking for is a population of cells in which 100% of cells are expressing your fusion protein. Culture conditions, passage, split rhythm, number, etc. of your selected cell type are critical for the generation of stable cell lines. For optimal results, we recommend following the cell culture recommendations of the supplier for the respective cell type. In general, to promote good proliferation and cell physiology, the cell line should be passaged 2 days before the experiment. Besides, cell passage should not be higher than 30 due to the possibility of interference with integration efficiency. Choose the G418 concentration. Susceptibility to G418 is different among cell lines and may even vary with cell passage numbers. The selection condition for your specific cell type needs to be determined experimentally. Determine the minimum level of G418 concentration to guarantee the minimum impact on cell growth. Note that the active concentration of stock G418 can vary considerably from batch to batch. Therefore, we recommend testing G418 sensitivity for every new batch. The final plating density depends on the specific cell type and the G418 concentration. We therefore recommend matrix titration of G418 and titration of cell number for determination of plating density in 196 well plate. Pre-plate 100 ml medium in each well of the plate. Add 100 ml of cell suspension containing 4,000 cells per well to the first column. After gentle up and down pipetting, carry over 100 ml to the next column, thereby diluting in a ratio of 1 to 2. Repeat this procedure for each consecutive column. Discard 100 ml from the last column after completing. The first column should then contain about 2,000 cells per well. The last column contains around 1 cell per well on average. Add 100 ml of G418 containing medium to the first row for a final G418 concentration of 1.4 milligrams per milliliter. Add G418 to the following rows in decreasing concentrations of G418 in steps to 0.2 milligrams per milliliter. For the last row, add medium without G418. Incubate cells at standard conditions. Analyze cell growth by microscope. In some cases, cell growth can also be observed by a change of medium color. If you observe cell growth after 10 days in the wells without G418, it is reasonable to assume that those cells can grow out starting as single cells. Choose the G418 concentration, which is just above the one that shows complete cell death, as the appropriate G418 concentration for selection. Transfection. For transfection, please follow the manufacturer's instructions for your transfection system. The important thing is to transfect the expression plasmid containing the target gene and the sequence for a drug resistance gene into your cells. We suggest setting a negative control of untransfected cells for selection. Besides, it is much better to check the transfection efficiency and integration frequency of your experiment with a GFP control plasmid. Cell selection post-transfection. After transfection, allow cells to grow and to express the protein for G418 resistance under non-selective conditions for about 24 to 48 hours. Use suspension cells directly or trypsinize adherent cells for analysis. Analyze for transfection efficiency 24 to 48 hours post-transfection by microscopy or Western blot of your target protein and positive control. Count living cells via trypan blue staining or other appropriate methods. Using a standard medium and the appropriate amount of G418 pre-tested for your cell type, plate cells in a 96 well plate with different cell numbers per well, in a volume of at least 100 ml per well. Depending on the cell concentration determined before, conduct several serial dilution steps as applicable. It is important to thoroughly suspend cells before seeding, but avoid harsh treatment by frequent pipetting. Incubate cells under selective conditions, and feed cells regularly with a fresh selection medium. Cell clones can be analyzed or further expanded as soon as cells in the non-transfected control wells have completely died. To help ensure that selected cell populations are clones derived from a single cell, another round of limiting dilution under selection is recommended. Analysis of stable cell lines. Once you have obtained resistant cell lines, you should expand the cells and assay your target gene compared with positive and negative control. You can detect the expression of fusion protein by an appropriate analysis method such as Western blot, microscopy, ELISA, as well as flow cytometry.